Hi, this is Stu Foreman. I'm going to be sharing some of the research out of our lab that has been done primarily by Ryan Fantasia, who's been working with me for the last couple of years. Um, sorry, the general area that we're interested in here is trying to gain more precision in understanding how etomidate binds to GABA-A receptors. And on the left, you see the molecule of etomidate. On the right is a structural model of GABA-A receptors, a little bit older structural model, but showing in red here the etomidate binding sites that are formed between the um, so-called beta plus, the bluish side chains, um, and alpha minus, the yellow side chains in the transmembrane domain of these pentameric anionic channels. So in terms of the hypothesis and the methodology, what we're trying to do here is to gain more precision using a technique called SCAMP, or substituted cysteine modification protection, that we've actually used for a number of years. And on the left is the sort of general model of SCAMP. We substitute a cysteine with a free sulfhydryl for an amino acid that we believe may be in or near the binding pocket of a drug like etomidate. We then use a cysteine reactive probe, in this case PCMBS, to covalently modify that sulfhydryl, which results in a functional change that we can monitor using electrophysiology. The protection part of this assay begins when we first bind etomidate to the site, and if etomidate is bound sufficiently strongly, it will prevent PCMBS from reaching the sulfhydryl and causing this reaction, thereby reducing the rate at which the covalent bonds and the functional effects of that change occur. And we can then infer that there's steric interactions. On the right is a close-up model of the transmembrane interface between alpha on the right and beta on the left, with a number of residues highlighted that were identified using PCMBS and SCAMP. But an important consideration here is that PCMBS is 9 angstroms long, which is actually longer than some general anesthetics that we think bind in this region of the GABA-A receptor. So to improve the spatial precision of the technique, what we've done is we've applied Alkyl-MTS reagents, where adding a single methyl group causes a change in length of about 1.3 angstroms, after correcting for dihedral angle. And the focus of our studies has been on this beta-3 M286 residue, because previous studies with the cysteine mutant at that position have shown that it has favorable characteristics for these kinds of studies. So here is some of the primary data and in alpha-1 beta-3 M286C gamma receptors. And to just elaborate a little further on the hypothesis, we hypothesize that when the modified cysteine side chain overlaps with the etomidate site, that we'll see 3 functional outcomes. One is increased GABA sensitivity to GABA that we see with PCMBS. The second is reduced modulation by etomidate, since we assume that there's going to be steric interaction between the side chain and etomidate at this point. And the third is that if etomidate were bound before the Alkyl-MTS exposure, that it should significantly inhibit the effects of that exposure. So here we're focusing on GABA sensitivity, but what you can see is with exposure to 9 millimolar seconds of Propyl-MTS, before exposure, GABA activates only about 2% of the maximum current. 10 micromolar GABA activates about 2% of the maximum current. After that modification, we have increased the GABA response at 10 micromolar about 3 to 4-fold, so a very large change. And that contrasts with Ethyl-MTS, which causes really almost no discernible change at all in GABA sensitivity. I'd also point out that shown here is how we also compare modification effects on etomidate modulation. So we're using a single sweep technique where we look at the current with 10 micromolar GABA versus 10 micromolar GABA plus 10 micromolar etomidate. So when we look at the summary slide for the GABA effects of modulation over a variety of these Alkyl-MTS reagents, we note that those that do cause significant changes all show that the changes are all dependent on either the time of exposure or the concentration of exposure. But most importantly, we see that ethyl and methyl MTS really cause no change relative to control exposures, which are to GABA alone. And we see the onset of effects at Propyl-MTS. So there's essentially a cut-on effect seen with the modification with Propyl that's not seen with ethyl. When we look at etomidate sensitivity after, or etomidate modulation after exposure to Alkyl-MTS, what we see here again for Propyl is a significant reduction in modulation. Looking in more detail, we find that the amplitude of the modulation goes down, but the etomidate sensitivity of these the so-called modified receptors is unchanged from wild type. And that's actually consistent with the idea that modified receptors are simply totally insensitive to etomidate, and the remaining modulation we see is due to unmodified receptors. And again, in the summary, note that relative to control, methyl-MTS causes no change. Ethyl-MTS actually causes a significant increase in sensitivity to etomidate. So that's inconsistent with what we expected for a modification that overlaps with the etomidate site. And again, the first modification that results in a reduction in etomidate modulation is that for Propyl, and the larger reagents do the same. This slide shows the effects of exposure to hexyl-MTS in the presence and absence of etomidate. So when there's no etomidate present, as you can see, the post-modification effects of both GABA and of etomidate are dramatically changed relative to the pre-modification effects. When etomidate was present during exposure to the same amount of hexyl-MTS, the change relative to pre is much, much lower. And over on the right is the summary for those reagents that cause significant changes in GABA sensitivity, and basically what we found is that addition of etomidate during exposure to these Alkyl-MTS reagents caused significant reductions in the effects for Propyl, butyl, and hexyl, but not for the other reagents. So in conclusion, these studies show a cut-on effect between ethyl-MTS and N-propyl-MTS with regards to GABA sensitivity changes, etomidate modulation changes, and the ability of etomidate to block these modifications. And these are persistent functional changes consistent with a covalent bond formation. So in terms of our interpretation of that result, this suggests that relative to the native methionine shown on the top here, the fact that the cysteine modified with Propyl-MTS is approximately 3 angstroms longer, this suggests that that the etomidate site is at most about 3 angstroms from the original, from the amino acid methionine, which is the native one. And our hope is that applying this technique at multiple residues in that pocket will help provide additional constraints on the location of receptor-bound etomidate. So I want to end here and just say thank you to the other members of our lab who have contributed to this research. And I look forward to talking with you and taking your questions at the meeting. Thank you.